Today, we have the great pleasure of having Dr. Wei-Sheng Cheng. He's a professor of environmental studies at UC Santa Cruz. And his research foci are on rhizosphere ecology, carbon and nitrogen cycles, isotope ecology, ecosystem responses to environmental change, and agroecology. He received his PhD in ecology from the University of Georgia, Athens. So thank you so much for taking time to come and talk with us. And I'm really excited to be here today. So thank you, Wei-Sheng.
Thank you. Thank you, Margaret, for the great introduction. Let me share my screen here. Okay. Thank you, everyone, for being here today. And hi, Tom. I saw your name there. I first thank the organizers, Margaret, Tarna, Lucy, and Han for organizing this and giving me the opportunity to talk about the rhizosphere and nitrogen, and especially gave me the opportunity to learn from folks here today. As you know, that I've been mostly doing basic research, so my knowledge about organic production is really, really limited. So whatever I say I'll present today, please treat it as food for thought and just to start a discussion on the complex issue or topic of the rhizosphere and nitrogen cycling. The word rhizosphere was first coined by a professor in Germany about 120 years ago. And the definition of the rhizosphere is the soil matrix inhabited and influenced by plant roots and root-associated biota, which is a generic definition. Many people using definition based on the kind of work they do. So for example, the spatial scale of the rhizosphere could span from, well, several order of magnitude, I would say, from as small as a few millimeters away from the root surface to as large as the whole rooting volume in the global land ecosystem. So in a global Earth system context, the rhizosphere is like a major hotspot for biogeochemical cycles. It really regulates all nutrient cycling and water movement across the whole globe. Let's get to the main topic we really want to talk about is the rhizosphere effect, excuse me. Just give a little narrative here. As we know that most of our textbook and research articles and data about rhizosphere or about the soil functions, such like soil organic matter decomposition rate, we all use soil samples. When we do that, there is an inevitable question. Does the rootless soil functions in the same way as the soil with roots? This is a major question that has been with me or guiding me for the past 30 plus years. So look at this slide. This is a real imagery, a real color using a camera buried to the ground with a clear plastic tubing. And so this is a piece of a very fine, fresh corn root. You see the root hairs all over the place. The volume of those root hairs can occupy a huge amount of soil volume. But when we taking the samples out without the roots there, you know the soil not gonna do the same thing as in situ situation setting. So let's define the concept called the rhizosphere priming effect by how we quantify it in this example, which I have done this mostly in past study in greenhouse or in growth chambers for many years now. So let's suppose we have pots, just the soil without plants in them. We call it unplanted control. But we keep the soil moisture and temperature at the same level as when we have plants growing in those pots. So that'll be the two bars here shown. Then we'll use let's say some kind of method. We can separate the carbon from the live plants versus the carbon from the soil organic matter decomposition, the CO2 produced by the two entities in the system when we planted plants, plants in the pots. Then we measure how much CO2 produced in these two pots or these two group of pots. The difference we call them is because of the Rhizosphere priming effect. That is the stimulation of soil organic matter decomposition simply by the presence of live roots. Okay. For studying nitrogen priming effect, we do not have to use an isotope necessarily, because nitrogen, we know, except in the case of a legume, all the nitrogen has to come from the soil. So we can do nitrogen budget to get at the effect of live roots on nitrogen mobilization, like the green texts show here. So we often use percent Rhizosphere priming effect for nitrogen. The N subscript P stand for the treatment with plant. The nitrogen was unplanted treatment, the control. So the nitrogen really standing for total mineral nitrogen in soil plus the amount of nitrogen taken up by plants. So this is together total mobilized nitrogen, available nitrogen. By doing this, we have to assume our total nitrogen recovery is 100%. That is to say, if your budget is not very accurate, you lose something in the measurements, then your priming effect may not be accurate or be off. All right, so that's how we do nitrogen priming effect very often. So Juji already mentioned in the last seminar, in general, the Rhizosphere effect is very variable. For carbon, for soil organic carbon decomposition, it can vary hugely. Some studies show that when you have live roots, it can change the decomposition rate of organic matter somewhere from sometimes you're getting a reduction of decomposition by 50% and to as high as 380% increase. That's nearly fourfold increase of soil organic matter decomposition because you have roots in it. All right, so it's hugely. For nitrogen also varies quite a lot. And Joji cited one study by one of my previous grad students here at Santa Cruz. But there are many other studies shows also highly variable. I'm going to present some results published in the literature later a little bit. Okay. And Joji also mentioned most of it we know about the priming effect based on results from pot experiments, either in greenhouse setting or sometimes we also bury pots in the field, but still there are pot studies. So there is some grain of salt in the results. How much relevancy, how much relevant of these results to the field setting is still a question. So this we've done a so-called meta-analysis, which is really we collected all the published results about priming effect. Then we would collect all the measurements reporting the quantitative level of priming effect, mostly based on carbon decomposition. Then we analyze is there any trend in the overall study of all published results. So this is based on 35, I believe, 35 publications and close to 200 observations. Basically, the overall average of all reported results for carbon decomposition is about 59% increase of soil organic matter decomposition because of the live roots effect. Then we also notice different plant groups tend to do different level of priming effect. With the published results, woody species like trees and shrubs tend to produce higher priming effect, but it's a smaller sample, 11 observations. Grassland plant species, somewhere in the middle, crop is kind of low, and other kind of species varies a lot. Okay, we also notice there's tend to be a separation of different soil texture. Finer soil texture, that is, soil texture have more clay and silt particles, tend to produce higher priming effect than coarser texture like sandy soil. And there is some indirect support of priming effect in so-called elevated CO2 studies. The kind of experiment they done is using very expensive technology called free air CO2 enrichment or phase experiment. So one publication summarized this study published in Science a while ago, saying the red text here I quote from the article, saying that including priming in ecosystem models captures increased decomposition rate with increased atmospheric CO2 concentration and results consistent with our finding. So basically saying that if they do not incorporate the priming effect on soil organic matter decomposition, their model cannot match with the actual data. There is a discrepancy. Only when they increase the K factor, which is here indicated in the bar graph, then their model can fit the actual data. All right, so that's a general introduction about rhizosphere priming. Let's get to our main topic. What does the rhizosphere priming do to nitrogen availability? All right, so this is one of my own study. It's a greenhouse experiment, pot experiment, many years back when I was at Louisiana State. So let me explain what this one does. So the insert is on the organic carbon decomposition, where the color, the blue, I mean the green and red and blue bars are on nitrogen. So we did both for nitrogen budget because we included a legume, soybean. We could use budget to get the nitrogen fixed by the soybean. That is the net increase above total nitrogen in the soil where we start with. We can also use the nitrogen 15 signature of the nitrogen fixed from the atmosphere as another way of estimating nitrogen fixation by soybeans. You notice the average is roughly the same, but the budget method gave much higher error bar than the N15 method. Okay, so the overall story, all right, so this on the left here, let me see, okay, this is what's in the shoots, in the background plant tissue. This is what's in the roots. The blue bar, I mean, the green bar is for soybean, the red bar is for wheat, and then the blue bar is for no plant control. You see this middle three is what's left in the soil in terms of inorganic nitrogen. And these two are nitrogen fixed by the soybean. So we take the nitrogen fixed by the soybean, fixation by the soybean out of the budget. And then we estimated how much soil nitrogen get mobilized by the plants compared to the no plant control. So the difference here is the Rhizosphere priming effect. You noted there, soybean tend to prime the soil nitrogen more than the wheat. The soybean getting about roughly 20% more nitrogen from the soil than the control. What's the mineral nitrogen available in the control? Where wheat only get a few percentage more. And as compared to the carbon decomposition picture, you see the middle bar is the wheat, the filled dark bar is the soybean. The increase of decomposition is huge. So the bottom line story here is priming on organic carbon decomposition may not be the same as affecting nitrogen availability or nitrogen acquisition. They're not necessarily linearly correlated. And there are also other studies we've done in the greenhouse. This one, we use 2 tree species. It's Fremont cottonwood and ponderosa pine, and 3 forest soil type. Well, 2 soil from forests, and the one from our local grassland near the UCSC campus. So there's total of 6 combinations, plus 1 that is the soil without plants. All right, so the figure here shows the y-axis is the gross nitrogen mineralization rate. The x-axis showing the priming effect of soil organic carbon decomposition. So there is a significant positive correlation. That is to say, the higher the soil organic matter decomposition, the higher the gross nitrogen mineralization rate. Let me explain what is the gross nitrogen mineralization here. We gave the word gross nitrogen mineralization. Really what we're saying is the cumulative production of ammonium from microbial activity or microbial decomposition of organic matter. So it's a total cumulative ammonium production. Where very often we use this definition in reference to net nitrogen mineralization. That is the net amount of ammonium left after the microbial immobilization due to microbial growth. Okay, so basically what we show here is the total microbial release of ammonium is correlated with root priming of carbon. Okay, and the same study, we did another data, I guess, gathering in a way we show that total nitrogen mobilized in the system, plants and soil, is logarithmically correlated with prime carbon. So that's the main trend of the data. However, there is 4 dots outlier, where we often call, they're not in the main group here. That's the treatment of blooded soil with ponderosa pine. They have pretty high priming effect based on carbon decomposition, but they're not related to nitrogen mobilization as the rest of the group. So what's the bottom line here is that most of the data show there is a positive trend between nitrogen mobilization by rhizosphere activity and rhizosphere priming of organic carbon decomposition, but there are exceptions. Okay, so let's just get started on this topic. So clearly, plants' live roots can modify the nitrogen availability in the soil. Now let's ask this question. How can plant root compete with microbes for nitrogen in the rhizosphere by giving mostly carbon-rich compounds? to the microbes. That's very often in the form of root activates. They are mostly carbon. So intuitively, plant roots seem to do something foolish for themselves, because when microbes getting carbon substrate, they will have a stronger competitive hand for nitrogen than plant could get the nitrogen. So what I'm saying here is there is something doesn't make intuitive sense if we say root activates could help the plant get nitrogen. How? So this is has been a mystery for me, and it took me quite a while to figure out maybe there's something going on more complex. We cannot use intuitive sense here. And we came up with a diagram like this. This is a complex diagram. Let me explain it. So basically, we're thinking there might be a different dynamic between the microbes. This is this box indicating the microbial biomass, the living microbial bodies in the soil. And this indicating the amount of plant-like roots. The middle is this pool of, say, available nitrogen for both plants and microbes. So let's assume we know for a fact, actually, microbial population and bodies, they grow and die at much faster rate. Some estimates saying that the bacteria living in the rhizosphere could die like a few hours after they grow in the rhizosphere. And where plant roots in the growing season, they take the nitrogen up and they don't return it back to the soil. They move the nitrogen to the above ground tissue for photosynthesis and other plant growth. So within a growing season, the nitrogen taken out by roots do not return to the soil. So that's why the flow is one direction here. But for microbial population in relation to the available nitrogen, the flow are two-way street. So they're taking up some of the nitrogen to grow. When they die and decompose, they release those nitrogen back to the available pool. If that's the case, if you use a simple mathematical calculation, if plant giving the microbe carbon or substrate have the microbe grow and die very fast, the net effect is that the plant will have a constant supply of available nitrogen. Then the plant root will win the competition giving long enough time. Do I explain that clearly, Margaret?
Yes.
I want to make sure. Okay.
But
so wait.
Yeah, I followed you. Yep.
Thank you. Thank you. So what we're saying is the growth and death rate of the microbe is the key in this rhizosphere interaction for nitrogen or competition for nitrogen. I'd like to add another loop called microbial loop in some ecological literature. The speed of microbial death very often related to faunal grazing. That is some animals living in the rhizosphere using microbial body of primarily bacteria and some fungi as their food. When they graze those microbes, they speed up the microbial death rate. And now we also know with recent studies, virus, the other agent mainly responsible for microbial death rate. So together, a healthy soil system should have very healthy food web structure, which often maintain a very high microbial growth and death rate. Okay. And if we put this hypothesis to explain the nitrogen competition between microbes and the roots in the rhizosphere, we could add the root to the diagram that published in the article by Dan Geisler et al. In 2010 in a review paper. Okay, so I borrow that without permission, by the way, stealing the diagram from them. Okay, so this also indicating for the flow between, say, mineral available nitrogen organic or mineral, available nitrogen resources to the microbial biomass, there is the two-way street, two-way flow, where within a growing season, all the available nitrogen to roots tend to be one-way flow. So basically saying that in this setting, plants can outcompete of the microbial activity for nitrogen if they have the microbe grow and die faster. And let's see, this is also from one of my own publications in 2009. We do have evidence, empirical evidence, support this hypothesis. So the 3 bar on the left is the rhizosphere stimulation of soil organic matter decomposition by wheat and soybean compared to the no plant control here. The 3 bar on the right is a measurement called microbial biomass residence time or turnover time, which is the inverse of turnover rate. The longer of the turnover time, the slower the microbial turnover rate. Okay. So without planting, the no plant control here, on average, the microbial biomass turn over one time in about 165 days. Okay, almost half a year, okay, 6 months. Where when we have wheat growing in the soil, the microbial biomass turnover time get to 61 days, 2 months roughly. So in other words, it takes 2 months of time have the microbial biomass grow and die once. Where when you have soybeans, that time dropped to 42 days, little less than 1.5 months. So basically you see the two indicating the faster the microbial turnover rate, the higher the priming effect on soil organic matter decomposition. So let's put this in a general perspective. If what we have proposed or hypothesized is true, the rhizosphere priming effect is primarily because the microbial biomass turnover rate are changing by the root activity, not a input from the plants. Then we could pretty much use it to explain some of the phenomena we know about. Let's say first, we know when the soil under warmer weather, they tend to decompose faster, and we tend to also get more nitrogen released. In that, I think we also know higher, warmer temperature tend to make the microbial biomass grow and die faster. That's we already know. That is based on observation that microbial turnover rate is way faster in tropical ecosystem than temperate ecosystem, even dramatically different than the Arctic system. So warmer temperature in the soil tend to not only speed up the decomposition, but also speed up microbial turnover rate. Secondly, we also know if a soil system have more soil organic matter, most of the cases, the microbial biomass is a relatively constant proportion to the soil organic matter, somewhere from 2 to 5% of the organic matter is made up microbial biomass. So therefore, if we have more soil organic matter, we'll have more microbial biomass, and very, very often we will also have more nitrogen release from microbial activity. Thirdly, when we have healthy soil, We also have healthy food web interaction, that is including all the necessary soil animals or soil fauna in the soil system. They tend to speed up the microbial growth and death rate or turnover rate. Therefore, you may also have more nitrogen release in that system. And the fourth phenomenon is we call the hotspot phenomenon. Now we know in the soil, they're not well mixed. The soil is very heterogeneous. Right? We all know that. If you dig up, you notice the different spot, different things happening. A common hotspot is the fine root and root tips. They are the globally known and most abundant hotspots. So therefore, we already show you that the microbial growth and death rate are much higher in the rhizosphere. There are other hotspots like earthworm burrows, animal casting, etc., or a big piece of a plant litter buried in the soil can also form another hotspot. Or compost, organic fertilizer, they all have hotspots phenomena. They tend to release more nitrogen. This is also because at the hotspot, microbial population grow and die faster for two reasons. One, there's more food, more concentrated substrate, so there's more microbe growing there. And virus tend to get the bacteria more easily when the population density is high. Similarly, soil animals grazing on those bacteria like nematode and other animals like protozoa, they also love high concentration of bacteria population. They graze them more when the population density is high. All right. Lastly, we also know when you have deeper roots, the lower horizon of the soil tend to have soil organic matter concentration is low, but nitrogen content is high. When you have root activate getting to those spots, you artificially can create a hot spot, and that tend to give the plants more nitrogen because the organic matter there tend to have more nitrogen relative to carbon. All right, so this is a 5-point summary on that hypothesis. We still need more support to firm up that hypothesis, but I think we have the initial indication that tend to be true. But I would like to warn everyone here or pay attention to the phenomena called immobilization. I explained that the immobilization of nitrogen is related to microbial growth taking up the soil available nitrogen, but now returning it back to the soil mineral nitrogen pool because they locked up in the microbial biomass. That will get us to this slide. This is a publication a while ago in Plant Soil. I'm a co-author of it. There is a summary hypothesis, which is the graph, I guess, Margaret was showing in the announcement, I think. So this is a hypothesis explaining how nitrogen immobilization may play out in the context of rhizosphere priming effect. The top, the two figures on the left, the top one basically shows a scenario when the soil system is dominated by nitrogen mineralization. That is to say, there's more nitrogen release in the system than microbial growth or fixation of nitrogen. Then, if that's the situation, more carbon input in the rhizosphere as activates or other carbon input from the roots will stimulate more rhizosphere priming effect. But if the system is extremely nitrogen limited, in other words, it's a immobilization dominated system, more carbon input from roots can result in negative priming effect. You're getting less nitrogen available. So different soil, different settings could differ quite a lot. To support what we've been discussing, there's a recent publication. Again, it's a collaboration between a few German scientists and here, Santa Cruz with us. We collected, published results, mostly within our groups, the German group and our group. And then we plotted out the priming effect based on unplanted soil as a percent of unplanted soil, excuse me. That is the x-axis. The y-axis is the nitrogen 15 isotope abundance as measured by a so-called delta value. So the higher the delta value, the higher the amount of nitrogen 15 in the nitrogen pool. The data we plotted here are the delta value from shoots, the top part, and from roots of all study we can collect. Generally, you can see the positive correlation. So let me say the top part shows the higher the priming effect of rhizosphere effect on the soil organic matter decomposition, the higher the isotope abundance of the shoots. Let me explain why this is important. The 15N abundance in the shoots is a direct indication that source of nitrogen is most likely come from microbial residues from the soil. It's because microbial residues has been recycled many times. They have a tendency, as the older the residue, the higher the nitrogen 15 content. So higher nitrogen 15 content abundance in isotope language, they are more likely come from the microbial residue. The other word called microbial natural mass. And so this is another support to our hypothesis. Rhizosphere priming are linked to microbial turnover rate. The higher the priming effect, the higher the microbial turnover rate. Therefore, the higher the N15 abundance in plant tissue. Okay, I'm using that in a reverse. And the line, the regression is even more significant for nitrogen in the roots. So therefore, we put out a diagram showing the recycling of nitrogen within microbial activity is the key for plant enrichment of N15. It's the key for rhizosphere priming effect. Now get a few more studies on this, then we'll finish. How am I doing on the time here, Margaret?
Yeah, you're fine.
Thank you.
Yeah, it's 10:42 or 12:42. Yeah, go ahead.
Okay, I'll speed up. Okay, so Judy showed this graph. I just stressed that the different plant species can do very different things to the priming effect. In this case, the three vegetables, broccoli, lettuce, and spinach, has a huge rhizosphere priming effect when they get fertilizers together. Okay, so where the two row crops show very different picture. The priming effect is way less, fertilizer not. Well, without fertilizer, have higher priming effect. With fertilizer, they don't, compared to the vegetables. And another study, again, Bill Drews, as a previous graduate student in my group, compared soybean with nodules and soybean without nodules. It's the same cultivar, except one will not grow nodules, so they don't fix nitrogen. What the paper shows is that nodulated soybean tend to have higher priming effect, but this figure doesn't not saying that. This figure shows priming effect of carbon and priming effect of nitrogen. Earlier part, when they are both low, they are linearly correlated. But when they get to some level, they level off. Or even, the efficiency of carbon drops, what we say. So there is this saturation level of carbon priming versus nitrogen mobilization. And another study, again, it's a collaboration with my Chinese colleagues. This and Australian colleagues. This study showed that phosphorus fertilization can also change the rhizosphere priming. Let me explain this here. The toolbar on the left is the control without plants. Then the toolbar in the middle is planted with ryegrass. Then the tool on the right is planted with clover. The red bar is the treatment fertilized with phosphorus. The blue bar is the treatment without phosphorus fertilization. You notice the switch when there's, let's see, did I make it wrong? Excuse me. My screen somehow, let's see. Yeah, so, oh no, I got it wrong. So the blue bar is the one. No, excuse me. The red bar is the one with phosphorus. Let me get this right. So with clover, they fix nitrogen. So nitrogen tend not to be a major factor affecting priming effect. When you fertilize them with phosphorus, the priming effect drop a little bit, but not as much compared to wheat, to ryegrass. See, ryegrass, when you fertilize with phosphorus, the priming effect on nitrogen drops tremendously. That's because we know when you give the soil micro more phosphorus, they immobilize more nitrogen and make the system nitrogen limited, extremely limited. So the priming effect on nitrogen get tremendously reduced compared to the treatment without phosphorus fertilization. So I've used this case as a example. Multiple nutrient interaction can be very important for what's going on with nitrogen in the soil. And again, the same first author did a study on multiple nutrients effect of rhizosphere priming. The conclusion of that article just published this year, showing that organically bound nutrients like nitrogen, phosphorus, and sulfur were closely connected with the rhizosphere priming effect on soil organic matter decomposition, where the rest of nutrients like base cation and micronutrients tend to do their own without really related to organic matter decomposition. And which this colorful figure shows on the left are the organically bound nutrients. Nitrogen, sulfur, and phosphorus tend to relate to overall organic matter decomposition. The nutrients on the right part of the figure, they are primed by rhizosphere activity, but not because of decomposition necessarily. They can be affected by rhizosphere acidification. They can be affected by some compounds chelating, some of the micronutrients, etc. All right, but not because of decomposition. So, in summary, the rhizosphere effect is pivotal for soil organic carbon storage and nitrogen availability and storage, and the dynamics of many other plant nutrients. But the assessment of the rhizosphere effect in field trials remain a challenge. And I would say thinking and investigating plant roots and their rhizosphere may offer some ideas when there are difficulties to solve some of the problems by looking at crop above ground or soil samples. And I'd like to finish by showing this beautiful art with an ancient wisdom. Thank you.